Thanks so much for having me today. I'm looking forward to lots of discussion afterwards, lots of feedback. I'm going to be talking about kind of our overarching goal to kind of provide a deeper understanding of human tissues. I think we all kind of have the longer goal of helping to inform clinical decisions as we go. And today I'm talking specifically about pancreatic cancer. And this work was really led by Faris Nowlin and Noor Shakfa in my group, but really enabled by a really collaborative team in the lab and without, and a lot of clinical collaborators as well. So towards this goal of really helping determine clinical decisions, a lot of the information that's used right now is in some ways quite simple and in some ways advanced or complex. A really basic decision is which tissue did a cancer arise in, and that will help determine what type of treatment those should receive. Pathology can really inform kind of the aggressiveness of the disease with grade or complexity, and is even the key factor that's going to determine this is malignant or non-malignant. And we've done a ton of work in genomics to really understand individual driver mutations. And even in some cases, this can inform in clinical decisions. In many cases, there are specific biomarkers. We also work in breast cancer, and for example, the presence of hormone receptors or HER2 will dictate if people should receive treatments against those. And in others, there's transcriptional profiles. So kind of what's the tissue state that we're looking at, and this has helped to define different types. And I think all this very deep information is helping to inform new treatments, and there has been amazing successes with increasing survival rates in almost every single cancer. One that continues to be a major challenge, though, is pancreatic cancer, where we're focused on here and hoping to make some big improvements. And major challenges can also be associated with pancreatic cancer, maybe why this is a big clinical issue. So for example, the tissue of origin is internal. It's really difficult to detect early masses or early tumors, and so these aren't ever found until more late-stage disease. The pathology is very unique in that there's a massive desmoplastic reaction. So in some cases, up to 80% of the tumor is actually the stroma and the fibroblasts and the immune cells. It's not actually the tumor cells themselves. And so it's hard to study the tumor cells driving this when they're only a small proportion. PDAC is actually considered quite a homogeneous genome. It's kind of known that there's very consistent 4 main drivers in the disease. Most patients have KRAS mutations, and then a large portion have deletions in these other drivers. There's no real known biomarkers that are out there or known for what treatment decisions should be made. And transcriptional profiles have done a ton of work, and these have identified kind of 2 extreme types: basal, which is thought to have worse outcome, and classicals, which have terrible outcome, just not as bad as basals, let's say. But this really only captures about 40% of the patients, and a huge portion of them actually are considered hybrid or mixed or of an indeterminate transcriptional profile. So this technical challenge with the pathology is something that we really think needs spatial technologies to address. This small portion of the sample actually being the tumor cells that needs to be separated out from the stroma to be studied. And this is also biologically very important, as these tumor cells are very ensconced in a very dense matrix interacting with lots of different fibroblasts. This can actually change their phenotype, the tumor microenvironment, and it can also control the immune response in specific locations. And so we really need to understand not just one compartment at a time, not just the immune, not just the tumors, but how all these fibroblasts, immune, and tuber cells are actually interacting and how they're working together to form a tumor. And so to get at this, we're kind of combining spatial technologies in multiple ways, using imaging mass cytometry, but also pairing this with other spatial methods to kind of capture more detailed information on a smaller subset of patients. And so to do this, our standard workflow is to kind of look through what's out there. A huge resource now is these cell tissue atlases, so we'll actually look at public single-cell RNA-seq. We'll use this to define targeted antibody panels that we're interested in. We've worked with the Campbell Lab to develop a tool called Cytomarker that will be discussed later today by Michael Gunick. And then we will use this to define imaging mass cytometry panels, and then we can study cohorts from our tissue bank on FFPE tissue sections to understand the cell content within these. We can map these back on the images and even look at how these different patients with different cell types responded or what their overall outcome, or look for associations with clinical data. We're then going to use other technologies to kind of dive deep onto these features that we've really profiled using imaging mass cytometry. So to summarize kind of what the overarching data set that we're looking at here is that we've actually combined 3 independent panels, one to get the tumor, one to get the immune, one to get the fibroblasts. So this is a combination of 3 40+ marker panels, 106 independent unique antibodies, and 20 markers that connect or overlap between the panels. We've captured almost 2,500 IMC images from our pancreatic cancer cohort of about 220 patients, so from multiple regions within every single tumor. From this, we can identify 170 different cell types. This can kind of capture the tumor heterogeneity, also very detailed immune cell content, the fibroblasts, very actually surprising detail in different types of endothelial cells, and even nerves that are present within these tumors. So the tumor-specific panel that we're looking at can be seen here on the left. This was designed to capture the different types of tumor cells that have been identified in pancreatic cancer, as well as new transcriptional profiles that we saw from the single-cell RNA-seq. This resulted in many, many different tumor cell types of great heterogeneity, but by using their co-localization and co-occurrence within these tumors, we could really summarize these down to specific phenotypes that you can see on the left. This nicely linked right back to the transcriptional profiles that we're interested in. We could see basal cells, we could see classical cells, but also further subdivides these types into a spectrum of different phenotypes that kind of span from basal to classical and break down these classical types. But looking at these cell types and how they're associated to clinical features, we could see things you'd expect. Basal cells are more present in basal type tumors that were identified by RNA. And then we could also identify relationships to things in the genome, such as ploidy or the lymph nodes metastasis status of these different patients. So kind of moving to the next imaging panel, we've taken a serial section, looked at the immune cell content, and within here we're looking at upwards of 50 different cell types. But what we could really define is that in the T cells, we could really break these down based on exhaustion markers that are present. Do they have PD-1 expression, CTLA-4, tons of TIGIT actually present in PDACs. We could identify kind of activation, a whole range of T cell subsets. On the myeloid side, we're also looking at TAMs, MDSCs, 4 different DC subsets, tons of different cells all going on in their location within these tumors. Finally, in the fibroblast, what we could really do is we actually were able to take 2 different classification systems present in PDAC, show how these aligned and merged, so both CD105-based definitions and MyCAFs and ICAFs, and also define the matrix content in these tumors. So if we're doing this by RNA, you can't see what actual matrix proteins are present outside the cell in the desmoplasia, and we could see a big difference between kind of a simple matrix that was present or a very complex matrix with other fibroblasts. And then a nice feature that we could add in is not just markers for the cells, but also their response, and in this case, biophysical response. Are they activating pathways that means they're under physical forces? So things like YAP or hippo signaling that was mentioned earlier, phosphofocal adhesion kinase, phospho-MLC2, and look at which cells are actually under a physical response or tension. And this allowed us to kind of break down in very detail the fibroblast contents across all these pancreatic cancer patients. What we were able to do is that if you look at the content of these tumors, we could define specific patient groups that are present here. And so what we could see is, but based on the majority of cells, we could identify classical tumors could break into 6 different types. It was really related to the expression of transcription factors associated with pancreatic lineage. We could also identify hybrid cells. These are both classical and basal, and this is a type of tumor. They're hybrid tumors. And also basal cell tumors that would be present or known 20%. Using the imaging mass cytometry data, we're able to break down these 7 different patient groups and see that there was very different outcomes in these patients, different survival. We're able to break down classical tumors into different survival and also see a big difference between these classical tumors and basal, as we'd expect, and even bad outcomes in the new hybrid subtypes and other subtypes that we're looking at. But if we looked at kind of the hazard ratios, or what was the risk associated with individual cells, it wasn't just being part of one of these groups. What it was is the proportion of dangerous cell types that were present in the tumor was really associated to outcome. So if you looked in the blue type, this is the one corresponding to these classical high tumors, the best outcome. They didn't really have an effect on the risk to the patient. What did is the presence of these kind of classical low cell types, those not having pancreatic associated transcription factors, and even hybrid cells. And then, of course, basal cells as well. So it was how much of the bad cell types are in the tumor was really related to what happened to these patients. So if you looked at the cell content in all 220 patients, here are the stacked bars. Each bar corresponds to an individual patient and the proportion of different tumor cell types in those. You could see that the blue tumor types way on the right, there's always an additional portion of these kind of dangerous cell types within them, and this was really most associated with what happened to these patients. So beyond just kind of having the proportion of the tumor cells, we have a very detailed readout of the different fibroblasts and the different immune cells. And so what you can see is the shape of the violin, which we still have a bit of a feel for, looks at the distribution of that cell type. Blue versus red is going to indicate kind of how this compares to average. So all the red spots that you're seeing are higher than average. Blue is less than average. And then the black outlines are going to identify those that are statistically significant within one patient group, not actually. And so there are ones that are high in multiple patient groups, but those won't come out here individually. And what you're looking at is kind of the blue is kind of the myeloid cell populations, red are the different T cells, and then we're getting into kind of the yellows and greens, which are different CAF types, into different blood vessel types all along the way. And what was unique is we can see that these types of tumors identified by the tumor cells also have unique fibroblast and immune cell environments. And what we can see is from these good outcome tumors down into the bad ones, there's a decrease in the amount of T cells that are present within the images. There was also different types of TAMs and different types of myeloid-deprived suppressor cells present in the different patient groups. And then into the bad type tumors, we could see new types of fibroblasts were really being enriched. Those with kind of a very complex, very matrix-dense CAF environment were present more in the basal tumors and the bad outcome classical tumors. So what we've seen is that there's tons of different CAF environments between these, but that these are actually quite complex and actually heterogeneous within the patients like the tumors. So the beautiful diagram on the right shows you that the strategy here was that we had 3 layers to this measurement, 3 IMC measurements, and what we did is we aligned the features of the tumors to identify different environments. And so what we did is we took advantage of the fact that these are tumor-sparse regions, and what we did is we aligned the tumor lesions across the 3 images in order to identify the stroma and immune cells that are associated with each independent lesion. And so from this, if you were looking at the images, you could see tumor cell markers on the left, a subset of the immune in the middle, and the fibroblast or matrix content in the right. And so with this spatial analysis, we could then separate out individual lesions that were present within one image and within an individual patient and see that these actually would have different tumor phenotypes, but also different immune and fibroblast content. So you can see kind of the purple lesion is different from this red and green mixed ductal structure that's here. You can see that the red and the red cells that you're looking at are the T cells, and the purple are the CD8s. Those seem to be physically associated specific with that duct and not this one, whereas the green MDSCs are more really enriched in this type of tumor. And at the same time, the fibroblast content is drastically different between these 2 lesions. And so really we needed to look the tumor type, the fibroblast content, and the immune content all in individual lesions is where the patterns aligned and where the actually biology of interest was. And so what we did is we used this information to kind of classify different types of environments. So in this case, the heat map, each column is indicating a different stroma or immune cell that we're looking at. And we could see that these were localized within 9 specific environments. So the top on the right that you're looking at is very immune-rich, immune-infiltrated, kind of towards the middle. Then we were looking at immune vascularized. And then towards the bottom, there were about 4 different types of fibroblast environments that were present. You can see that there were many fewer cell types present in those groups that are kind of down to the right, but their abundance can still be quite different. So the bars here are looking at how many different types of cells were present within these individual environments. And if we looked at then where these environments were in the different types of tumors, every type of tumor contained every single type of environment. So these environments could be present all at the same time within individual patients and in every type of tumor. But we could then kind of uniquely break apart the risk associated with having different environments. And so as the amount of immune suppressive environments or kind of complex ECM desmoplastic-like CAFs increased, we could see that those are increased with a statistically significant hazard ratio here on the right. So further, as I mentioned, we know that these all coexist. So what are the relationships between these different environments within a patient? And so looking at these 9 different environments, we could see how often they coexisted with others. And what this could kind of break down to is that there were certain tumors that would have more of these stiff biophysical environments and also at the same time have ECM-rich. And these were less likely to be associated with immune-infiltrated ones, whereas there was kind of a MyCAF-related population that would coexist within tumors that had big T-cell influx and immune cell content. So looking at our 7 tumor cell types, we could then identify, if we looked at different locations in these tumors, how much does the stroma environment change? Is this that I can sample any different region and I can call the same environment, or do these drastically change? And what was interesting is that the different types of tumors had different spatial heterogeneity. So along the top are kind of our transcription factor high subgroups. We could see that they were much more homogeneous, whereas this transcription factor low is the one that had this red dot out to the right, was kind of the more spatially heterogeneous immune environment and more spatially heterogeneous CAFs. Or at the very bottom, this basal cell type, this is kind of the highest, one of the high-risk subgroups that's known. You can see there's a blue dot way out on the right side. It had a very spatially heterogeneous fibroblasts, but not as much in the immune. So these tumor types, we know they have different environments, but they can also have different spatial heterogeneity, different variation within their environments. So far, all of this has been really looked at in these TMA cores, looking at these 4 sampled regions from a patient. We are always interested in what's happening across all the area of this tumor. And the limitation here is the amount of time it really takes to kind of image these things. So using standard IMC, we'd be rasterizing with 1 micron spots. This means that it can take multiple days to do an individual whole slide, but we can do multiple TMA cores all at the same time. So using this newer tissue mode, what it's actually doing is increasing that spot size. And so instead of 1 micron spots, we're taking 5 micron spots. And this means that the resolution of the images is lower, but that the spot that's being taken is 25 times larger. And then we can rasterize that tissue way, way faster. And so that what that means is instead of taking these little spots, we're actually able to take multiple whole slide tissues every single day. So running 24/7, this allows us to really capture many whole slide samples off of these. And so what we were able to see is that when we looked at the whole slide samples from a lot of these patients, we could see things that across all the data set, about half the patients are quite homogeneous. You can see the middle and the one on the left, they all seem to be kind of one tumor type. And the other half started to get increasing heterogeneity. So the tumor type on the right, you can see that there's kind of a blue tumor type and a red and yellow one. And so that one had multiple types of tumors within it. So this all confirmed what we'd seen on the TMA, but provided a huge area for all of these tumors to look at this heterogeneity. So within this one patient of interest, we could actually see the 2 standard types. So on the right, you're looking at in cyan as a classical type tumor, and the red cells that you're looking at the left are the basal cell types. So within this one patient, you have what the clinicians want to classify into 2 bins. They're both present within the same patient. Beyond the tumors, we can then see where are the immune cells responding. So now we're super zoomed out. This is a whole slide. So each little tiny dot that you're looking at here is an individual cell. And what we could see is the red cells here are CD8s, and we could see that they were kind of focally enriched in specific little patches. And then the green cells are MDSCs, kind of suppressive cell populations. And you could see that there are some regions with a suppressive immune response, some regions with the immune. And then further, we can go and look at what's the fibroblast content in those same locations. And we can see that there is a relationship to different types of fibroblasts associated with the classical type on the tumor on the right, the basal type tumor on the left. And that this is also related to the immune cell type. So if you remember, there was different T cell hot MDSC regions. There are also regions that the immune cells are completely absent. And those seem to be related to kind of these blue FAP positive type regions here, that there was actually an absence of most immune cells, but there were still some rare TAMs in that location. So at this point, we'd really nicely profiled, had a detailed understanding of these little microenvironments, that there's tons of them in every single patient. But we really wanted to kind of dive deep and see these features that we see across all the patients, what's happening in these. And so what we're able to do is to take these, we're able to take these specific regions, and we're able to move to a serial section. And using the image from the IMC, we can guide where we're going to do laser capture. And so from this, we were able to actually capture 275 different individual environments. For this, that every type of the 9 environments and every types of the tumors that we were looking at, we captured between 6 and 12 different regions, all from more than 3 patients. This means that each capture is between 20 and 100 individual cells, and taking this and introducing it into a mass spec system that has been built up for kind of small amounts of samples with our collaborators at the Lunenfeld in the Gingras lab, we're able to get between 3,000 and 6,000 proteins from every single one of these small environments. And so in this case, we're not looking at transcriptome, we're actually looking at what proteins are present, and we know the cell content of these because we use the IMC to inform where to capture. And so this provided super detailed information about what was happening in the environment and associated tumor lesions within each one. But we looked at this across all of our environments and all of our tumor cell types to identify pathways that are present within these and then different cellular functions in these environments using the proteomics. And so to confirm and to investigate the cell relationships to this, we also linked our mass spec imaging data to public single-cell RNA-seq. So we confirmed all of our cell types from the PDAC that we're interested in, and we're looking at how do the pathways that we see in the mass spec, how are these activated in different individual cells from single-cell RNA sequencing. And what was really nice is that what we could see is that things that you'd expect. In environments that were super enriched for classical type PDAC, we're calling single cell populations and PDAC specific transcriptional programs. In basal type cells, we're calling basal transcriptional programs. And then into the immune environments that we're looking at, we'd see a major enrichment in pathways associated with T cells or MDSCs in our suppressive environments. But beyond just having that these are capturing these specific cell types and CAFs as well, we're also able to call all of these different pathways. And in some cases, these pathways are cell type specific. The environment has more cells, therefore it has pathways associated with that cell type. But it can also be pathways that are specific to a certain environment, and that the spatial information from the mass spec is really helping to distinguish different things happening in different locations. So for example, we could see metabolic activity that would be present in certain types of tumors, and then different metabolic activity in certain environments that were present within this. We could also see different glycosylation pathways, and some of these made a lot of sense. Mucin-type associated glycosylation was happening in the epithelium and the ducts, whereas more production of the matrix and different types of different matrix production for glycosylation were associated with the environments we were pulling associated with CAFs. Of interest, multiple development pathways were also being called in this. So something that we're seeing is that goblet-type cell differentiation was present within our tumors. This is actually a stomach cell, but what's being seen is that this type of differentiation can be associated with a damage response. And so this is something we're seeing in our lesions, but also is being pulled out of the single-cell RNA-seq. And then an interesting one was a kidney development pathway being associated with our fibroblast programs. And there was also multiple fetal-type single-cell RNA-seq signatures that we're seeing in our mass spec. So also in these immune environments, we could then pull independent signaling pathways. So some environments would be enriched for a specific pathway, and we'd help to identify IL-10 signaling seemed to be present in kind of different types of fibroblast environments, and that RUNX3, which I have never worked on before, seemed to be really related to what was happening in different immune cells within the immune-infiltrated environments. So overall, the strategy here has been really to deeply understand all the cell content that's present within a whole bunch of 220 samples, to look at very detailed samples, but then also to look at very large areas to confirm our findings and to look through these. And then from this, we were able to identify our individual regions, laser capture these for very deep mass spec analysis of these individual pathways, identify the cell content and organization and different activities present within these. And what I didn't have time to show you today is that what we've also done is we have whole genome sequencing from all of these patients. So very interesting upcoming work from this is that the cell organization that we're looking at here is actually associated with specific genomic aberrations, which will be very exciting as these are all kind of thought to be genomically homogeneous. And then using tools such as Stable, which is going to be discussed as well today, we're able to take the multi-omic data and identify which features are actually associated for predicting outcomes in these patients. And nicely for us, it really prefers the imaging mass cytometry data. So across all of these, the conclusions and where we go from here is that kind of new information that's coming from this is that in these pancreatic cancers, about half of them are one type of tumor, half of them are mixed, multiple types of tumors within them. We can further subdivide the current basal or classical classifications to get about 7 types of pancreatic cancer. These have different immune environments, they have different outcomes, and that some of these immune cell types that you'd expect are related to outcome. They just are happening in very localized events within the tumors. But more T cells is definitely a good thing. More suppressive cells is not. There's very diverse fibroblast populations within these. These kind of do exist in spatial organized patterns, and the basal tumors that we're seeing seem to have more heterogeneity, different types of CAFs in different locations. All of these little individual environments can be related themselves in their quantity to patient outcomes, and they do have different pathways activated within these different environments. And what's now coming, and I'm happy to show anyone that is interested to talk about it, is that these features can be associated to specific genomic aberrations, and that using these in predictive models can really let us identify that the rich information coming from IMC and coming from these environments can be very useful for prediction, and that the single cell data has a lot of potential for that aspect. So all this work was led, as I mentioned, by Nurk Shakfa and Ferris Nowlin, but really dependent on a big collaborative team, collaborators in Kieran Campbell's lab and Claude Gingras's lab. Very tight relationship with the Pancreatic team at Toronto General Hospital, Steve Gallinger, Jennifer Knox, Fayez Nota, and Grania O'Kane, really driving the clinical questions. And all of this was really possible because they've spent a ton of time putting together this very detailed cohort that we were allowed to contribute to. So really need to thank the environment and the team, and I'm looking forward to questions from all of you.